So guys, I'm talking about destruction of red blood cells. If you want to discuss the destruction of red blood cells based on site, you could have a red blood cell damage that is basically what outside the blood vessel. All right, a damage of the red blood cell could be outside the blood vessel. All right, or it could be inside the blood vessel. All right, so picking the outside the blood vessel now to discuss, and outside the blood vessel could be in organs other than the blood vessel. Do you understand? So extravascular hemolysis is a site of red blood cell destruction. That's what we're going to actually um, discuss in this lecture. So you see, extravascular destruction is basically could be occurring in the spleen mainly, but it can occur in other organs too, like the liver. Okay, so talking about this, so that basically red blood cells are phagocytosed and degraded by macrophages in the spleen. It could be in the liver or it could be in the bone marrow. Okay, so now within the macrophages, the hemoglobin is broken down into its constituents. All right, and the constituent is basically the hem ring, which is a porphyrin ring with iron in its center. Then we have the globin chains. All right, could be alpha and the beta. All right, so now basically these globins are broken down into amino acid, which are then used for protein synthesis. Okay, porphyrin ring of hem is oxidized by what microsomal hem oxygenase, producing biliverdin and releasing the ion. Okay, so this is just talking about the process. Now next, ion that is released is then exported into plasma through ion channels. All right, where it binds to transferrin, which are basically what the transport proteins. Ion can be stored within cells as ferritin, and in ferritin the ion is bound to storage proteins known as what the apoferritin. Okay, so ferritin is oxidized and degraded to form what hemosiderin. Hemosiderin within macrophages can be stained with Prussian blue. Also biliverdin is what reduced by biliverdin is reduced by biliverdin reductase to unconjugated what bilirubin, which is then released into plasma. All right, so unconjugated bilirubin is transported by albumin to the liver. Okay, the whole story continues on this last slide. Now the unconjugated bilirubin is conjugated to hepatocytes. The conjugated bilirubin is excreted into bile canaliculi via the ATP energy dependent transporters. Right, bilirubin is secreted with bile salt into the intestine. Here the bacterial enzymes degrade it into what? Urobilinogen. All right, so about 90% of urobilinogen is broken down into urobilin and stecobilin. However, about 10% of urobilinogen is reabsorbed, and most of it is what? Taken up by the liver via the portal vein, and the rest will bypass the liver and is excreted in urine. Okay, so this is just basically how extravascular hemolysis of blood takes place. All right, I'll be seeing you guys in the lecture. We'll be talking about the intravascular one. Bye.